now we want to talk about the problem of how does the cell know where the introns are? In other words, how does it know where to splice? And in this sense, a cell faces very much the same problem as a film director who has shot thousands of hours of film and now is overwhelmed with film and needs to then make a final movie. And so traditionally with film, before the digital age, the film director would go through the film frame by frame, find exactly the place where he or she wanted to make a cut, and then there would be a splicing machine that would literally cut and splice the film back together to make the final version of the movie. And in cells, that's exactly what happens, because of course introns are made up of individual nucleotides, and the nucleotides are very' like the individual frames in a film. So introns have the following structure. The 5' end, or the beginning of the intron, is many times called a splice donor, but splicers tend to call it the 5' splice site. And it, in 99% of genes, is signified by a GT as the first two nucleotides of the intron. And then much further downstream, actually close to the other end of the intron, is an adenosine called the branch site. And we'll be talking about what that branch site does in part 2 of my lecture. And then at the very end of the intron is the AG, which is the last two nucleotides of the intron at the 3' splice site, or the splice acceptor. Now, I've told you that some introns are up to 400,000 nucleotides long, and certainly this amount of information content is not enough to even signify a small intron. So there must be additional sequences. And so in addition to these universally conserved nucleotides, there are consensus sequences in all of the introns. And so here are the consensus sequences for budding yeast. And these are called logos, motif logos. And the height of the letter tells you how conserved that site is. So for example, here you can see the GT at the 5' splice site. And so there, in addition to the GT, there are several other nucleotides that are highly conserved, and a few that are less conserved. At the branch site in yeast, there is a very highly conserved TAC-TAAC sequence. And then upstream of the 3' splice site, there's not too much conservation until you get right to the 3' splice site. So there's additional information in these so-called consensus sequences. Surprisingly, in humans, so if you remember, yeast only have about 250 introns, and they generally are short. They're all less than a couple of hundred nucleotides, whereas human introns are much, much longer. And the surprising thing is that the human consensus sequences are less conserved. So you can already see that, because you can just see that there are lots more letters down here than there are up here, particularly for the branch site, much less conservation. And then for the 3' splice site, there's a little more information, because there's this so-called pyrimidine track here that is a bunch of C's and T's upstream of the AG. Let's take a look back at that model of the dystrophin gene that we looked at. So if you remember, here's my long 99-foot rope. And when your eyes are looking at this rope, probably what they're drawn to are the exons, the little pieces of color, and not the rope itself. And so you can actually think about a human gene as islands of exons in the middle of oceans of introns. And so very much like a map of the Pacific Ocean, if you're going to identify the islands, you're going to look at the islands, not at the ends of the oceans. You're going to look at where the islands begin and end, and not where the ocean begins and ends. So let's think about how that might be applied to a human gene. What we now understand is that the way that introns and exons are recognized in human genes is through what we call exon definition. And that is that even though the introns are of highly variable length, going from about a few hundred nucleotides up to tens of thousands, hundreds of thousands of nucleotides, The exons tend to be very much uniform in their length. The average exon, as we said, was 123 nucleotides, but they vary in length from about 25. There's a few shorter. There's exceptions to every rule. But in general, 25 to 300 nucleotides is the size of an internal exon in a human gene. And so what the human splicing machinery looks for is a good match to the consensus branch site and 3' splice site, followed with 25 to 300 nucleotides by a match to the 5' splice site. And so this then defines an exon. So finally, I'd like to leave you with my take-home messages. The first is that eukaryotic genes contain introns. Not every eukaryotic gene contains an intron, but certainly in humans, the vast majority contain introns. These introns facilitate the evolution of new genes, and by allowing for alternative splicing, they allow us to greatly increase our proteome complexity. And higher eukaryotes utilize exon definition to define splice sites, because the exons are like islands in oceans of intron. And then finally, many human hereditary disorders are caused by mutations that affect exon inclusion or splice site choice. So this ends this part of the lecture. If you're interested in learning something about the machinery that removes the introns, load up my next lecture, which is spliceosome structure and dynamics. Thank you. Hi, I'm Melissa Moore from the University of Massachusetts Medical School and the Howard Hughes Medical Institute. And in this lecture, I'm going to tell you about some work from our own lab on spliceosome structure and dynamics. So as we saw in the first lecture, eukaryotic genes are split in that they have expressed regions or exons up here and introns. And the introns have to be removed. And in my previous cartoon, I've just used the scissors and tape. So in this lecture, we're going to be talking about what is the nature of those scissors and tape, and then how do they actually do the reaction. So first, let's talk about how the splicing actually happens. And it happens in two chemical steps. In the first step of splicing, the branch site, so we talked about that, that's a conserved adenosine in the intron near the 3' splice site. The 2' hydroxyl of the branch site attacks the phosphate at the 5' splice site. And then that generates a lariat intermediate, which has a 2', a 5', and a 3' phosphate all coming off of that adenosine. And it releases the 5' exon. Now that 5' exon doesn't float away. It's going to be held onto by the splicing machinery, which we'll be talking about in a moment. The second step of splicing, so here are our intermediates in the reaction. This 3' hydroxyl that was generated in the first step on the 5' exon, it now attacks the phosphate at the 3' splice site, and this now kicks out the intron and ligates the two exons together. Now we know that this is the chemistry, and that these occur as single transesterification reactions. The two steps of splicing are catalyzed by a large complex in the cell called the spliceosome. The spliceosome is arguably the most complicated macromolecular machine in the cell, as we'll see in a moment. And the spliceosome consists of 4 major chunks, large pieces, subunits that need to come together with each round of splicing in this complicated dance called the spliceosome cycle that we'll talk about in a few slides. But what I want to show you here is that the pieces of the spliceosome, the major pieces are these components called U1, U2, the triple SNRP U456, and the 19 complex, or the NTC. Here we are at the spliceosome, back at the spliceosome cycle, and we're going to look at this a little more closely because I'm going to be telling you about some experiments we've recently done to test this cycle. So in the first part of the cycle, at the beginning of spliceosome assembly, U1 SNRP interacts with the 5' splice site, and U1 snRNA actually base pairs and recognizes the 5' splice site. Similarly, U2 snRNA base pairs with and recognizes the branch site consensus sequence. And U2 snRNA, when it joins the U1 snRNA, forms A complex. E complex stands for early complex, and then A, B, and C, as we'll see in a minute, were just named by where they ran on a gel. Okay. So A complex comes first, and then the next big chunk of the spliceosome that comes in is the triple SNRP, U4, 5, 6. Once the triple SNRP is there, that we have B complex, then there's a structural rearrangement where U1 SNRP is actually ejected, it's kicked out, and somehow things are majorly rearranged such that now U6 snRNA is interacting with the 5' splice site. In another rearrangement, U4 SNRP is kicked out, and then the 19 complex comes in, and now we have the catalytically active spliceosome, where the first step and then the second step of splicing occur. Once the second step of splicing is complete, the spliced product is released, and the spliced out intron leaves with the rest of the spliceosome. That splice machinery has to disassemble, and then it's reassembled anew on each new round of splicing, so ergo the spliceosome cycle.